Good morning, everybody. Sorry for the momentary delay. It's always a bit of a challenge when you're dealing with technology, both for in the room and for the folks participating online. So on behalf of the Kawartha Lake Stewards Association, my name is Tom McAllister, and it's my pleasure to welcome you to this morning's spring KLSA meeting. It's so nice. I haven't gotten over it yet, the fact that we're all back in the same room again. It feels like it's been so long, but it's so nice to be able to see people and spend a little bit of time together. So we'll be going from now until noon today, and I think we've got a great presentation that you'll really enjoy. So I'd like to begin with a land acknowledgement. We would like to acknowledge that the greater Peterborough area is in the Mississauga, Shinnecock territory, in context of the Rice Lake Treaty 20 and Williams treaties. In keeping with KLSA's mission, we wish to emphasize the efforts of Indigenous peoples as water protectors. There's much that we can learn from Indigenous knowledge of the land and what is needed for a more sustainable and regenerative future. A little bit of housekeeping this morning, as always. This meeting is being recorded. We have people participating, obviously, in the room. We've got a bunch of people online in Zoom as well. For the folks who are online, we would ask that you input your questions, just type them in. Bob will be keeping an eye on the screen, and we will introduce your questions when we receive them or shortly thereafter. If at home, you'd be kind enough to please mute your mic and video except when speaking. In terms of mechanics in the room, we will have members of the Kawartha Lake Stewards Association board will be coming up and talking about their individual programs. Just giving a heads up, if everybody who's coming up here to speak could come around that end of the table, then they won't trip over the cords that are at this end of the table. We just don't want, health and safety paperwork is a killer. Okay, so next slide. In terms of the meeting agenda, we'll start with the chair's report, and then we will talk about the contest winner this year. I hope you've had a chance. If you haven't, please do take a look at the display board at the back with all of the contest participants, some wonderful photographs. We'll talk about this year's lake water quality report, and we'll talk to our programs, our ongoing programs at KLSA. Then I will introduce Jeff Berthelette, who will come up and talk about a program that he's leading this year. Then we'll take a break, and after the break, Nolan Pearce will come up and speak, and then we'll wrap it up. So I'm going to go to the next slide, please. Chair's report. I'm going to turn it over to Bob with both mics. This is Bob Bailey, the chair of the KLSA.
Thanks very much, Tom. I was saying to Judy beforehand, there's more tech involved in this because we are doing this hybrid. That's why you're seeing us flail around a little bit, especially me flail around a little bit at the front here. More tech involved than at the coronation. So bear with us right now for folks on Zoom. If I could get a thumbs up that you can actually hear me, that would be great. From Hayward, thank you. And now you can mute your video, please. And yeah, make sure all of you are muted video and audio. That really helps with our bandwidth. So welcome, everybody. As Tom was saying, it's so great to get together in person again. We had a great in-person AGM.
Excuse me, somebody is not muted.
Sorry,
there's a lot of interference.
I'm just getting, I think it's voices from Zoom in my
Brian
head.
to mute himself. Thank you, Brian.
Anyway, excuse me. Yeah, we had a great AGM last fall, and great to be together again for a few reasons. One, it's a beautiful day in the Kawartha Lakes. I think you have to agree. Two, the Leafs are still in the playoffs. Luckily, we're having our spring meeting early in May. Three, I guess we officially have a new king in a couple of hours, if it hasn't happened already. I know my neighbor, Janet Duvall at Lower Buckhorn, couldn't be here because she's a staunch royalist. So I guess we should all sing God Save the King. I think, no, okay. Just a couple of things I wanted to mention. I wanted to start out by recognizing fellow board members. You've already met Tom, our esteemed vice chair, Tom McAllister. And if I call your name, fellow board members, if you wouldn't mind standing up so people recognize you, see you, at least the people who are here in the room. So Ed Leardom is our treasurer and past chair, and he keeps our money in order, our bank account straight. Sheila Gordon Delane is our secretary. And as Tom will talk about in a few minutes, so she keeps us on the straight and narrow with respect to governance and minutes and agenda, all that good stuff. She's just been an immeasurable help to me over the last few months as I've taken on the role of chair. So welcome to Sheila. And as Tom will talk about in a few minutes, she runs a crack team that produced these incredible annual water quality reports that hopefully everybody's grabbed a copy of. So good to see you here, Sheila. Jeff Chalmers is a past board member, recently past board member. I think Jeff's here. Yeah, I saw Jeff earlier. Carol Cole is a present board member. Welcome, Carol. Mike Dolby, another past board member, recently passed. That sounds kind of creepy. Mike's a very vibrant, recently passed board member. Leanne Green, great to see you, Leanne. I've never actually seen Leanne in person before, so wonderful to see you. And I know Daryl Cotton's here. He was helping us out, another board member, and Jackie Milne at the back there. She organized, she did all the ticketing and watched out for scalpers and things like that. So, and Kim Ong is here somewhere. And Brett Traguno texted me this morning. He's really sick, I guess non-COVID sick today, so he couldn't make it, but works for Kawartha Conservation and another valued board member. So recognition of all of them. Yeah, as Tom was saying, you're going to hear about our programs today. We've got a couple of great guest speakers. And as I point out in my chair's report and in the annual water quality report, you know, it's amazing what this organization does as a steward society. And a lot of it is you. So, I mean, I've been on a few volunteer boards, and trust me, the work that our board members do is incredible. But really, the volunteer beyond the board, the volunteer force that we have out there for these programs you're going to hear about and more is just phenomenal. It's more than I've ever seen in any kind of organization. So kudos to you, members beyond the board, and obviously folks that can't be here today. So you're going to hear about those programs. The only other thing I want to mention before we get underway is we had this note from Tanner Liang at Kawartha Conservation. Tanner actually gave one of our talks at our last spring meeting, and he was just wanting to let us know and our members know that Kawartha Conservation is launching a harmful algal bloom safety campaign next week. And those of you who, I think everybody or most of you have heard about harmful algal blooms, the blue-green algae blooms and toxins, which can be super dangerous, happen then. And a bunch of really interesting research going on about them at Queen's and some at Trent and a couple of other places. But anyway, Kawartha Conservation's launching this campaign next week, beginning this coming Tuesday, May the 9th, and running till the end of June. They'll be sharing information and resources on social media in collaboration with the health unit to promote public safety and community awareness of HABs. And so we'll be sure to tag that media in our social media, including our Facebook site and such, so that you can connect with that if you're interested. So having said that, I'm really anxious, looking forward to the presentations today. So I will, I think I'm turning it back to you. So we'll do this very elegant moving of the conches. I guess there's more than one time.
I hope that we'll manage to move the mics back and forth more smoothly than the Leafs move the puck. Anyone who's still cheering for them this late in the year, I'm miles ahead of you. I was at the Barbers the other day, and I was saying about the Leafs and how they always fold. And I said, they haven't won since '67, the last year of the 6-team league. And my barber said, I wasn't even born then. I was born in '69. So it really makes it makes you you really feel your age. Next slide, please. Martha, we want to congratulate you for the cover photography this
A
year.
little closer. We got to be on the
Oh.
all the media.
Oh, I,
All the media.
Paparazzi.
Congratulations
Thank
on,
you.
on your photographs this year. It's an absolutely beautiful cover. I hesitate to say it's the best ever because I don't want to put any of the others down, but it is a really striking photograph.
Amazing.
And congratulations. We've never had someone in our photo contest win first and tie for second. So obviously you're spending far too much time on the water.
The truth.
But sincere congratulations. Thank you so much.
Thank you.
Next slide, please. And these are we didn't have we had a first place. We didn't have a second and a third place. We actually had a tie for second place. You can see the one of the water lilies there is by Dave Morley on Chandos Lake, and the other one is the Narrows by Martha once again. So congratulations. And as I say, if you haven't had the opportunity, I would really encourage you to take a look at the wonderful photographs. There's just so much to savor in this part of the country. We're very fortunate, and the photographs do a great job of showing that. Next slide, please. Okay, the annual water quality report. Bob has already talked to it, and you've already seen the cover, and many of you have already had a chance to contribute or to review it. We've got lots of copies at the back of the room. I would ask all of the lake associations who are here today, please take copies back to your members. We've also got some great copies of former water quality reports from previous years. And if you'd like to pick those up, there's some sort of the older copies of the aquatic plants guide and so forth. There's lots of material back there. Please take it with you and enjoy it. There's lots of great information in there. This water quality report, as always, is also available online on our website, so you can see it there as well. Next slide, please. Okay, so now we're going to go around the horn where the various project leads within KLSA talk about their programs. And as luck would have it, the first programs up will be spoken to by Bob Bailey. Bob, over to you.
Okay, so reports. So I'd like to talk a little bit about two projects we've been involved in for many, many years. The first is the E. coli program, and the other, which we oversee, we manage. The other is one that we really facilitate our members taking part in. That's the Lake Partnership Program, Lake Partner Program. So the E. coli program, for the last few years, it's been run admirably by Cindy Lee in collaboration with Mike Dolby. And this year, I'm overseeing it with my partners, Daryl Cotton and Mike Dolby. So this is, as I mentioned, it's a KLSA program. It's E. coli sampling 5 times a year, starting July 4th, ending September 5th. And there's about 20 or so individuals or associations who take part in this, and each of them sample from 1 to 8 or 9 sites, and each of those sites in their lakes are sampled, as I mentioned, 5 times a year. We're doing things a little bit differently this year. I've contacted folks who have done sampling in the past and getting the information from them with respect to how many sites they plan to do this year. And I will be getting the sample bottles from the lab that we have analyze the E. coli samples. I'll be delivering the bottles to the various volunteer testers in June. And Mike and Daryl and I will be revising the instructions, including making a new little training video later this month, which will share the links for that for the volunteer testers. But basically, it's the same regime as I mentioned: 5 sample dates, and the volunteer testers will deliver the samples to the SGS labs in Lakefield following sampling, again, according to the protocol, according to the instructions that we'll share with you. But that part's working basically the same as last year. The value of this program to KLSA and our members is immeasurable, and you'll see a report on that on the program written by Mike in the annual lake water quality report, if you pick one up later, if you have one. If you're not taking part right now, we always encourage new volunteer testers. We don't cover all of the Kawartha lakes right now. So please contact me. You see my email up there, and there'll be a link to that on our Facebook page and website. Please contact me. There's still time for you to be part of this program this year. So any questions about that from folks? Oh, I'm sorry.
Does anybody do
Where's Mike? Okay, maybe we'll talk afterwards, okay? Because I can check that. I've got all the coordinates. One thing about it is we don't ask people, because of the nature of the sampling, so it's E. coli sampling, and we share the results with people. We share the results, as I mentioned in our report, on a kind of a lake basis, but we don't share the specific coordinates of the sites that are sampled, for reasons that I'm sure you can understand with respect to privacy and all that kind of stuff. So, but yeah, for sure, if you are interested as well, you don't just have to email, just check in with me either at the break or after the meeting, and we can see if it makes sense for you to take part. Always love to get new volunteers there. So the Lake Partner Program, again, long-standing program that we've been involved in. And I believe, Mike, you write up stuff based on available data from the Lake Partner Program in the Kawartha Lakes area. But it's important to realize this is not a program that we, in any sense, run. We help, we help our members participate in it if they want our help. But the main thing we do with this, and you can see the Lake Partner Program, which I should have mentioned before. So it involves taking water samples, a little bit more persnickety process, that's a scientific term, than the E. coli sampling. And it results in analyses of phosphorus levels in the water sample, chloride, calcium, and also water clarity is done because you do what's called a Secchi disc reading. You drop down, if you haven't done it before, you drop down this black and white disc, should have brought one. You drop it down and see when it disappears. And that sounds incredibly crude, but believe me, I've been in limnology for 40 plus years, and it's remarkable what amount of research and knowledge and management has been based on that very simple disc as a measure of how clear the water is. So anyway, those data are collected in May of each year. And the main thing that we do is, what's now called scrape the data. So Mike has done a great job of looking at the data. There's several, I think on the order of 20 or so, or maybe even more than that, 30 Kawartha lakes, 44, that our members do, sampling that at least people in the Kawartha lakes do. Mike looks at the data from the Kawartha Lakes area, but data from lakes across Ontario are available. This is a community science program that cuts across the province. And I put this long convoluted link there to their data search site. I'll have a shorter link available on our social media. So because it's fascinating. It's fascinating for me, and I think for you to do as Mike does. He has fun every year looking at the data that have been collected at sites in the Kawartha Lakes and beyond. So again, it's a little bit late to get involved in that program this year because there's a process involved in getting sort of signed up with the Lake Partner Program. Again, this provincial MOE program. But if you're interested, please contact me. And I'll help you out to get involved in the future, or maybe just to look at the data from your lake or other lakes that you might be interested in.
Bob, we have a question for you from the webinar. Can you hear us?
Okay, so Brett Traguno, as I mentioned, he's ill today, so he asked me just to say a few words about the what we call the climate change monitoring program. And this is, again, it's a community science endeavor where it's entering the third year of a 3-year pilot program that I think we're going to continue. It's so valuable. I've been involved in a little bit of climate change research lately. And the one thing that really comes out as a strong need is you got to have data. If you want to know how things have changed, and Nolan will talk about this later in his talk, if you want to know how things have changed, you really got to keep track. You got to do that regular, I'm just going to say boring monitoring. It's never boring for those of us who have done it, but you got to have that base of data to understand how things might have changed when we get to 2030, or when we get to 2040. So it's not necessarily, this 3-year program is not going to have the smoking gun when it comes to climate change effects on Kawartha Lakes, but it's going to be the basis, and Nolan will show you a bunch more data. It's going to be the basis of judging how things change in the future. So what happens here at a bunch of lakes, and you see squiggly little line there, and you'll see lots of those in Nolan's talk later, showing how the oxygen level changes as you start at the top of the lake, the surface waters of the lake, and you drop down to the bottom. And I've had a peek at Nolan's slides, and he'll say much more about this much more elegantly later. But short version is that, when the oxygen declines in those lower, deeper waters, that makes things tough for a lot of things that live there. Changes the chemistry and all that sort of thing. But the bottom line, all organisms, all animals anyway, need oxygen to survive. And so a change or a depletion in the oxygen down there is important. So we keep track, and I think the latest count was we're into about 8 or 10 lakes, where, again, volunteer community scientists, like Mike is one, and I do it in Lower Buckhorn. Jeff, and anybody else here who takes part in this program. And so they go out, and it's usually, I think, about mid-August, right, Mike? Sometime before the end of August, kind of after the middle of July, in that time period. And they go to a deep part of the lake, and they send down an oxygen meter. And this oxygen meter, it's on a long cable, 30, 40 meter long cable. So you're taking the oxygen and temperature readings every meter as you go down. So you get a picture like this, and that's like a baseline. It tells you what's happening in that lake that year. And again, critical to have that kind of information for monitoring or assessing the effects of climate change. So those are the 3 programs I wanted to talk about. Any questions about the climate change monitoring? Yeah, Jeff. Thank you.
We also have in picture there is one of the loggers who attached to 5 Trent boys through the system.
Right. Yeah. And
Year round data prevent severing from down to 15.
yeah, thanks for the reminder, Jeff. So yeah, temperature monitors, which record, if they'd only had them when I was a grad student, but they record constantly data about temperature. So lots of folks have those. I think at the end of their docks, they deploy them, sort of from May until October, give or take. So we get that picture of what's happening in the near shore. And then Jeff's a veteran of, because he's a great scuba diver, among many other things. And so he's part of the team that goes out and deploys these temperature samplers on the Trent Severn buoys. And so they're in the water through the entire year. And so getting that picture, again, is super valuable, and that's part of the program as well. So if you're interested in it, again, get in touch with me, and we can get you as part of the team. I can't finish without recognizing Mike Dolby. A couple of years ago, he really wanted to support this, and I happen to have a lab at Ontario Tech University, and Mike arranged a donation so that we would have a second dissolved oxygen meter to sort of do the sampling that community scientists do in this period for this program. So kudos to Mike. Anything else on that?
The third at Stony Lake years ago, and I've been Stony for probably 5-6 years.
Right. So between, and it's been a great, it's great community science, but there's actually something in that sharing around among organizations and people that I think is really valuable to the research.
Robert, can you hear us on the webinar?
Yeah.
We have a question.
Yeah, is that the Graham Raby?
The one from Trent.
Yeah.
Yeah, I think it's the Stony.
Okay, yeah, Nolan will probably Nolan's a postdoc from Trent, part of that group, so maybe Nolan pick up on that later. Thanks. Anything else? Okay, I'll turn it over to my colleague Kim Ong to talk about natural edge. So you have to put that on your.
Okay.
I'll let you do that.
Perfect. I think I got it. All right.
If you could stand over here. So.
Am I? Oh, okay. Well, hopefully everyone can stand out. Good morning, everyone. My name is Kim Ong, and I am part of the board of directors. And I'm really pleased to talk about the Natural Edge program in person. It is a program that we do in collaboration with Watersheds Canada. It's their program that they have set up, and we are their delivery partners for the Kawartha Lakes area. And basically, this program, the goal is to restore shorelines. So we look for landowners in the area, people who want some help with restoring their shorelines with native plants. And there's a lot of reasons that people might want to do this. It helps reduce erosion. It, of course, makes the shorelines prettier. It prevents you having to mow your lawn if you have a lot of grass there. And it really overall just helps the health of the shorelines. And also keeps so that's actually a big reason that a lot of people join as well. So we've been doing this for a couple of years now. We've been kind of in pilot stage, doing about 5 to 10 sites per year, and we're trying to ramp up now. So we're aiming to do 20 sites over the next couple of years per year, pending some funding. We've been in a little bit of a hiatus for the past season because we were looking for this extra funding, but we have committed to doing 10 sites this fall. And currently we are looking for landowners in the area who would like help with their shorelines. So we basically, we come to your shoreline, we assess it, we talk with you about what you would like to see on the shoreline, so shrubs, trees, flowers, and then we go away and we make this personalized planting plan for you. We go over it, and then we actually buy all the materials on your behalf, bring them to your site, and then if you want, we actually get a bunch of volunteers to actually come out and help you plant. So it's about a 6-month from beginning to end kind of process. And yeah, it's really, really valuable. It costs $250 for you, and we match that funding. If you have a really big shoreline, it's maybe a little bit more, but it's usually enough to get the planting going. And yeah, really, I know there's a lot of associations out here right now who might be able to get the word out to your members about this program. It's really, really valuable. So please come see me afterwards. My email is kim.ong@klsa.info, and I will be here. Or you can email our general line as well, and Carol will direct the emails to me. But yeah, we would really love to see another 20 sites over the next couple of years planted. It's really fun, and it's really nice to see. It takes about 3 years for the shoreline to fill in, but then you have this great native garden in your front yard. So I'll just stop and see if there are any questions. You stick try it? Okay. Oh, do I need to scroll down here? Okay, yeah.
We
I
do
don't
have a
wanna mess with it.
question on the webinar.
Whoops. And just the picture that we had on the screen there is just one of our sites that we did in this past fall. And you can see that we brought, I think this is quite a large site that had been really affected by the derecho. And so we had about 18, I think, volunteers come out and help plant. And this massive site took about 4 hours to get 300 or 400 plants in. Okay. Any questions in the room? Yes.
So it's a great volunteer at the municipal level. I know. Shoreline protection. And the county have regulations about having naturalized switchboard. I think for the top of the municipal folks and maybe the last or the mayor is like, because well, they're great, but it's all the way down. They can't do on.
Yes. Reaches. But yes, just to repeat the comment, we have a comment saying that there are some municipal bylaws coming in that kind of prevent kind of hardening of shorelines with the armor stone, for example, and instead would encourage naturalization of shorelines. And so the question was, have we been in touch with municipal offices? We haven't, but thank you very much for that suggestion. And yeah, just another reason to really maybe join this program and contribute. Was there anyone in the audience who wanted to comment on that? Yes, please.
I think we're probably going to be doing something like that. I'm not allowed to break right now, but we are all interested in making our shorelines as good as possible. Probably taking into the water. So I think that is coming in our.
Great, thank you. Okay, thank you.
Robert, are you there at the computer? Can you hear us on the webinar?
Good. Morning, folks. I'm Daryl Cotton. Good. Weeds. When you think of the word weeds, you're probably thinking of something negative, frustrating, undesirable. Everything in the water, weeds. I think that's the general perception of many folks. Admittedly, when I was looking for a cottage in 2019, I was one of those people. My family are nature lovers, the fish, the turtles, birds, Love them all. Didn't want anything to do with the weeds. We needed a clear waterfront. As I began to learn more about what was in the water, my perception started to change and became a lot more mindful. So to me, this really resonated. I think without the plants within the water, which I was perceiving everything as weeds and undesirable, there wouldn't be much life around. In fact, probably the water would become overwhelmed with algae and really undesirable. So the objective here is to start educating folks more on aquatic life, the plants within the water, leveraging the checklist that is now published in our report that's available at the back, and to create a bit of interest in what's below the waterline. So we're going to be running a contest, a scavenger hunt of sorts, and we're going to have prizes for the person that identifies the most plants and the most unique. So it's the overall number and the number of unique. It's going to be run June 1st to September 1st. And we're going to be leveraging the iNaturalist app. If folks are familiar with this app, it's a great resource for collaboration and for general enthusiasts to connect. And that's available online. And we'll also be leveraging Facebook, where we're having weekly posts, plans of the week. Also, we'll be running a leaderboard to just keep the contestants abreast of how people are doing. And, you know, I think the ulterior motive here is just to engage the public on the value of those plans. So we'll continue to build upon it as we move forward. But hopefully we can contribute to changing perceptions. Any questions there?
That link to the iNaturalist, is it up yet?
Not up. It'll be up in about a week. Hi there.
Can you send the link out through the?
Yeah, we'll email out the link and we'll publish as well onto the Facebook site. Did anything else come in there? Yeah. Hi there.
Are you actually going out yourself and doing any studies or anybody doing the research?
Independently?
Can you repeat the question?
Yeah. So the question is, are we going out ourselves and doing research? So, well, the way the contest is running and the way that the iNaturalist app works is that it's a community-driven initiative where individuals are identifying plants or any creature and then taking a picture and adding information to the iNaturalist site. And then specialists within the community get to comment on it and verify what's been found and provide further information. Based upon the community contribution, a database of information is built. We didn't have immediate plans to do our own analysis through this, because I guess the strength of the community probably is a lot more comprehensive, covering all the different layers.
He can't win the contest.
Yeah. But from my perspective, so I'm on Buckhorn Lake and I've had my own challenges in the past and misconceptions. I'm certainly participating from my own perspective, to educate myself better on what is in the waterfront in front of my property. And of course, to just further my own education.
But it's linked to climate change and the differing and the water quality and salinity and all that sort of
It's
stuff.
all, yeah.
It's all interconnected, yes.
So we develop a database for a period of time. So that's what that was going to be my inquiry as to whether either you're doing it for this year, next year, the year after, kind of thing. See what is changing.
Yeah.
Because our lake has changed like this. Hugely.
So the feedback is building upon a database of info. Carol?
So that's Barbara from Dog, and I get exactly what you mean. That is sort of the idea, is to track over time what there is in all the lakes, because as you know, it does change based on climate. So that all of it is together, but for sure,
Even one lake, say, a northern lake.
Right.
That corresponds to our.
Okay,
Yeah.
good. All right, thank you.
Okay, so my topic is actually, was going to be done by Brett, but since he's not here, I will speak to it. Oh, sorry. No. Okay, is that better? All right, I'm Carol, and aquatic plants are one of my passions in life now. For all of you that have been around a while, you've probably seen our 2009 aquatic plant guide. And so what happened with KLSA is, as interest have developed with aquatic plants and we started to view things a little differently, we thought it was time in 2023 to do an updated guide. There's plants that weren't in the Kawarthas at that point that are now going to be in our guide. And just a lot of sort of a philosophical change towards plants. A lot of our previous guide was how to control your plants. This will be how to get to know them and respect them and understand their role in the environment, which is really important. And as Barbara pointed out, is going to be increasingly changing and challenged over the next few years with climate change. So we have done a little contract with a grad student from Trent University, and she's going through and updating the guide for us. And we are hoping to have the guide ready and sitting at the back of the room when you come to our AGM in October. So that's about it. And the checklist is in the back. So if there's anything, if you're wondering what you might be able to find, there's a great checklist right in the back of our guide. So check that out. And that's the 30 most common plants in the Kawartha Lakes. So.
Thank you very much, Carol. I'd like now to introduce Jeff Berthelette. Jeff is going to come up, and he's working with the Ontario Federation of Anglers and Hunters on their invasive species project, and he's going to give us a brief update on some of the work that he's doing. Come on up, Jeff.
Awesome. Thank you so much. So first and foremost,
Just so they can see you.
hi everyone at home. Thank you everyone for, especially Carol and the KLSA for having me here today. Very grateful to be here. And shout out to an amazing report. It looks awesome, great information. So yeah, my name is Jeff Berthelette. I'm the outreach liaison with the Ontario Federation of Anglers and Hunters Invading Species Awareness Program. It's a bit of a mouthful, but basically within the Invading Species Awareness Program, it's a partner collaboration between the OFAH and the Ministry of Natural Resources and Forestry, where we work with targeted outreach and awareness to advocate against preventing the spread of invasive species. And so you may know us with our hotline, our 1-800-563-7711, where we collect reports of invasive species, things that we have get reports on them. We input that data into EdMaps, share that data with partner organizations to come up with and develop management strategies. And so my job as the outreach liaison is to manage the Water Steward Program. And so the Water Steward Program, it's actually only a couple of years old, but we're really happy with the progress that we've made with it. Just last year alone, we have interacted with and outreach to over 1,200 boaters and anglers. So that's 1,200 people that now know how to do aquatic invasive species checks, what to look for with aquatic invasive species. And obviously, if you don't know, invasive species are a severe threat to our aquatic ecosystems. The International Union on the Conservation of, hang on, I have it written down here. International Union of the Conservation of Nature in 2015 designated that invasive species are the second biggest threat to the loss of biodiversity. So it's obviously something that we're all very concerned about. Invasive species like water soldier, Eurasian milfoil even round gobies, zebra mussels, we're all familiar with the impacts. And so the water steward program just works to advocate against preventing the spread, providing the public and water enthusiasts with resources and the ability to recognize, identify, and remove aquatic invasive species from their boats as they take them out of the water. Now, one thing that we're trying to do a little differently this year, because I just came into this role a few months ago, we're really trying to empower and work with collaborating and outreach and networking and stuff, because thankfully we've worked really hard to secure a good amount of funding and resources that we can share with our partner organizations. So with that being said, if you guys, anyone here needs help preventing or advocating or doing any kind of outreach to prevent the spread of aquatic invasive species, let me know, because it is my job to work with all of you. And if you need resources printed, if you need extra set of hands at any kind of event, let me know. We've got a lot of manpower and resources that we can share. And ultimately, we're all fighting the same battle here to prevent the spread of AIS. And so with all that being said, outside, I've got one of our boats with me. And afterwards, during the break, I would invite all of you to come out with me and we can go over how to conduct an AIS check together. So yeah, if there are any questions about the water steward program. Oh, I see a couple. Yes. Absolutely. Of course. So the question was, what kind of signage do we have available for making the public aware of the invasive species? So we have put up hundreds and hundreds of signs all across Ontario, basically, with information on this preventing the spread of invasive species and how to identify them. I'll give you my card, and easy peasy, just tell me how many signs you want and we'll send you some. I'm sorry? Yeah, of course. So usually what happens is, lake associations or municipalities, they'll ask us for signage. We work with a lot of different partners as well. And then they just say, hey, can we get some updated signs? Yeah, absolutely. Here's the hardware as well. And also, again, we have the resource and the manpower to go out and help install these signs as well. Yes. Yes, that's correct. So because my job is the outreach liaison is just to focus on the water steward program, the Invading Species Awareness Program in general has a multitude of different programs within it. My colleague Brooke Schreier runs the wild boar surveillance program. Rob McCowan, he's another one of my colleagues. He works on management strategies with water soldier. So the Invading Species Awareness Program covers a multitude of different invasive species and their management processes. But my job is just to focus on aquatic invasive species and outreach and awareness. Yeah, of course. Thank you, everyone. Yeah.
All right, well, thanks for that introduction, Bob. Really happy to be here today. Normally, when I give talks, I'm lecturing to a number of students at Trent. So I usually start with some sort of question to make sure everyone's awake and paying attention. And since it sounds like we were all watching the Stanley Cup playoffs last night, I will start with a question here as well. So just by show of hands, I think this first question is going to be a simple one, but can you tell me if lakes are important to you? So everyone's raising their hand. Great. And now my next question is, are the important things that lakes provide or the things that you think are important about lakes and value under threat? So pretty much the same number of hands. I expect a little less, so I just put probably here. But my final question is, are the 900,000 Canadian lakes, equally important and equally threatened. Oh, we got a good class here today. So I've answered no here. So we know that lakes are really important. They, provide a number of goods and services, cultural, social, recreational, economical, environmental, the list goes on. We know that many lakes are threatened due to various stressors. We heard about some already. And climate change as well. But we also know that all the lakes across Canada are not equally important. And we know that these stressors differ from place to place. So while it's critically and urgently important that we protect our lake ecosystems, we also have to think about the regional context when we do this type of work. So this psychedelic map here aimed to just do that. And I thought it'd be nice to share with you today. So what these researchers did is they went across Canada to thousands of lakes, and they quantified 3 attributes of those lakes: its threat level, its ecological health, and that lake's socioeconomic importance. And then they mapped out these 3 attributes using primary colors, where the mixture of these colors indicates kind of high levels of multiple attributes at once. So I just want to focus in on the Kawartha Lakes and, quote unquote, Ontario's cottage country. And if we look here, we see this kind of purpley blue color in this region. And what that means is that the lakes in this region are of really high importance to a lot of people. They're in generally good ecological health, but that they are highly threatened. And if we add to that, this region is kind of right on the north bound of some of our kind of established environmental organizations like Ontario's conservation authorities. So they tend to receive a little bit less oversight than other regions here. But we do have to applaud all the great work by the Kawartha Lake Stewards and other stewardship organizations to take on some of this work in terms of better understanding and safeguarding these water bodies. So at Trent University, we also want to help address some of the threats to the Kawartha Lakes through both research and some long-term monitoring. And one of my interests is in dissolved oxygen loss in deep water lakes, as you saw from the title. So that's what I'll be talking a little bit about today. And it falls under the nutrient pollution category here, but I'm going to touch on a little bit about climate change. So a few summers ago, you might have been drinking your morning coffee on the dock and saw this article in the CBC titled "Bad News for Fishing, Climate Change is Sucking Oxygen Out of Lakes," study suggests. The study that this news article is referring to was published in the journal Nature, titled "Widespread Deoxygenation of Temperate Lakes." And what these researchers did is they collected long-term monitoring data from over 300 lakes across the Northern Hemisphere. And with that data, they looked at trends in surface water temperature and surface water dissolved oxygen concentrations. And what they found was that for the majority of these lakes, they saw the surface water temperatures were increasing over time, as you can see by that tall bar on the right. And within those lakes, they also found that the surface water dissolved oxygen concentrations were disproportionately decreasing over time, as you can see by the yellow portion of that right bar. So this article created a lot of buzz and got a lot of attention, but it's actually a really simple finding if you know a little bit about limnology. So for my class here, here's my limnology primer. So oxygen gets into lakes through gas exchange with the atmosphere. Gas exchange is a physical process whereby oxygen moves passively into and out of lakes through diffusion. This gas exchange is driven by wind and water turbulence, but this process is an equilibrium process, meaning that there is a maximum amount of oxygen that water can hold, and we call that upper limit 100% dissolved oxygen saturation. So you can imagine this as if you had a child or a grandchild who was making themselves a cup of hot chocolate. They wanted to add those couple extra scoops of chocolate milk powder into that hot chocolate. It doesn't actually dissolve in the water, it just settles out to the bottom of the mug. So the degree to which a substance dissolves in water is called solubility. And in regard to dissolved oxygen or oxygen, solubility is highly dependent on temperature. So if we look at this graph here, I'm showing temperature on the x-axis, the amount of dissolved oxygen that water can hold on the y-axis at 100% dissolved oxygen saturation. And if we look at this trend line, we can see this big decrease as temperatures get warmer. And what it shows is that because of dissolved oxygen solubility, colder waters naturally just have a greater capacity to hold on to oxygen and dissolve more of it into those waters than warm waters do. So if we're seeing increasing trends in surface water temperature over time, we must see decreasing trends in dissolved oxygen over time. So this figure here is another figure from that Nature paper, and we have on the left plot here on the x-axis, we have our temperature trend, so the increase in temperature per decade, and the x-axis on the right plot, we have our change in oxygen concentration per decade. And don't worry too much about the y-axis, it's just kind of the frequency of lakes in their data set. If we just focus on the red distributions here for the surface waters, we see that there is indeed an increasing trend in temperature over time, and we see that decreasing trend in dissolved oxygen concentrations. But if we look at the deep waters of these same lakes, we see this decreasing trend in oxygen concentrations, but we see no trend in temperature. So the oxygen is declining in these deep water lakes, but it doesn't appear to be related to any changes in water temperature. So what's going on here? So this is my last little limnology primer before I start to show you some data. But I think you already know what thermal stratification is. If you've ever gone swimming in the lake and you felt that cold water by your feet and that warm water at the top, you've experienced thermal stratification. So as water warms up in the summertime, the surface gets warmer, the bottom stays cool. And it is that difference in temperature, or that temperature gradient, actually prevents the mixing of those two parcels of water together. And what that does is it creates a barrier at that depth of thermal stratification, which cuts off those bottom waters of the lake from the atmosphere. So when thermal stratification sets in, those bottom waters now have a finite capacity of dissolved oxygen. There's a set amount that's there until the temperatures get colder again. We move into the fall. The temperature of the water column is uniform. It allows for full mixing throughout that water column and reestablish that connection with the atmosphere. This same sort of thing happens in the wintertime as we get lake ice forming on the lakes. It's going to create that barrier. It's going to cut the waters off from the atmosphere. There's going to be a fixed amount or fixed supply of oxygen in that lake until that ice melts and we move into the spring. The water temperature is uniform again, allowing for mixing and reestablishing that connection with the atmosphere. And we also know that these bottom of these lakes are home to many different organisms. And as Bob mentioned, many of these organisms require oxygen to breathe. So if there's a fixed supply of oxygen at the bottom of these lakes during the summertime when they're stratified, the activities of these organisms can actually start to decrease the amount of oxygen within that lake. In particular, all of the bacteria and microbes that are living in the sediments at the bottom of these lakes really like to consume that oxygen and decrease it over time throughout the summer season. And consequently, if we go out and we measure oxygen concentrations in lakes in the midsummer, so this is August 2021, we see profiles that look like this. So it's a little bit of a mess, but if you follow right from the top of the graph, we have depth on our y-axis and we have our dissolved oxygen concentration on our x-axis. We see that at the surface of the lake, we have high oxygen concentrations, but then as we get deeper and deeper into the water column, our oxygen concentrations get lower and lower and lower. But if you look at this graph, you see that there's a whole bunch of squiggly lines everywhere. So this is for, I think there's about 35 Kawartha Highland lakes here. We see tons of variability among these lakes in terms of their oxygen concentrations in these deep waters. So this raises the question, well, what makes one lake lose its oxygen faster than another lake? And it's this thought that kind of sparked or generated some research questions for our group. And the first is, what are the rates of deep water dissolved oxygen depletion in the Kawarthas? And then expanding on that, what are the factors that drive differences in these deep water dissolved oxygen depletion rates among lakes? And have these rates changed over time? We then wanted to address what are some of the ecological consequences of this dissolved oxygen depletion. And finally answer the question, is dissolved oxygen depletion a concern regarding the sustainability of the Kawartha lakes? So to do this, Bob already mentioned in his little intro there some of the ways we can do this with some handheld sensors. In our lab, you know, my supervisors fortunately have lots of money, so we were able to purchase one of these high-resolution vertical profilers, and this instrument can take measurements of a number of different variables, including dissolved oxygen, but temperature, conductivity, salinity, chlorophyll, at a resolution of 8 measurements per second. So as we lower this instrument down through the water column from the surface to the bottom of the lake, it is continuously taking the measurements of these variables, and we get this really high resolution data. But these vertical profiles, they've been collected for decades. They're a very common practice in limnology, especially those temperature and dissolved oxygen profiles. And they're very routinely used in broad scale lake monitoring. But the catch is that we typically only collect these profiles once during the year, and that typically occurs in August, later in the summer. And while these data are really, really helpful and really informative, they're kind of restricted to an individual lake basis. It gets a little bit more difficult to compare lakes or compare these profiles among lakes due to differences in lake morphology. So, for example, if we had a small lake and a big lake, that bigger lake would have a greater amount of that initial fixed supply of oxygen than that smaller lake. So to really understand how fast these lakes are losing oxygen over the summer season, we need to take at least 2 profile measurements during that period of summer stratification, and we need to merge that with some geographic data on lake volume and benthic surface area in particular. And if we collect all this data, we can kind of produce a graph that looks something like this. Again, we have our dissolved oxygen concentration on the bottom, our depth on the y-axis. Here we have a secondary x-axis at the top, which is lake volume, shown by that kind of curved dotted line. And our 2 profiles of dissolved oxygen collected in June and July. With these profiles, you know, specifically with the temperature data associated with them, we can determine what the depth of thermal stratification is. And then we can actually take our dissolved oxygen data, sum up all the oxygen that's there in that volume of water below the depth of stratification for June. We can do the same thing for July, take all that dissolved oxygen data, sum it up for that volume of water below stratification for that time period. We can simply subtract those two sums, divide it by the time that's passed, and we end up with a rate of oxygen loss. That rate of oxygen loss can then be standardized to the size of the lake. So how much surface area is at the bottom of your lake. And once we do that, we now have a standardized rate that we can compare among many different lakes throughout the region. So in the summer of 2021, we went out to 8 lakes every 3 weeks from May to September, and one time in February. And we went out to Big Cedar Lake every 3 days from April to December. We selected these 8 lakes such that they were heterogeneous in their lake morphology. Some were bigger, some were smaller, and that they differed in their summer dissolved oxygen concentration. So some of the lakes tended to have more oxygen in the summertime, some of them had very little oxygen in the summertime. And then we used all these repeated profile measurements that we took and the data on their lake morphology to calculate those dissolved oxygen depletion rates, as I previously described. And this is what we found here. So a simple figure, but it is, I did find it interesting. So the first thing that we noticed on this figure is that there's 5 out of 8 lakes right clustered in the middle that all have pretty much the same dissolved oxygen depletion rates. But if you know these lakes, they look very, very different from each other. They differ greatly in size. They also differ greatly in their summer dissolved oxygen concentrations. So it's interesting that they have similar depletion rates. The second thing that we noticed is that there is quite a large range between the minimum and the maximum depletion rate. So these kind of preliminary findings just made us more interested in, well, what is driving these rates of dissolved oxygen depletion among these lakes? So why are these lakes looking different? But I'm sure many people here who volunteer and collect water samples and do profiles can attest that just to get these 8 data points, it was a lot of work for us. We had to go out, all the time and collect these things, bring stuff back, process data. So it was quite tricky. And the 8 data points really isn't enough. There's not enough data here to really answer our question in terms of what is driving differences among these lakes. So I had a thought. And the thought was, using our knowledge of limnology and some of the things I mentioned earlier regarding regarding dissolved oxygen solubility and temperature, could we predict what these dissolved oxygen depletion rates are just based on one dissolved oxygen profile collected in August? So could we leverage our broad scale monitoring data that we have for many, many lakes to calculate these same rates? That way we have this big robust data set of the entire Kawartha lakes, and we can begin to answer some of these questions. So if you recall thermal stratification and solubility, for any given temperature of water, we can actually calculate how much oxygen is in that water at 100% saturation. The other thing we know about lakes is that when summer stratification sets in, the temperature of those bottom waters of the lake don't change very much throughout the summertime. They stay, relatively the same temperature as they were, as they warmed up to in the spring. So with our one profile that we collected in August, we know the temperature of the bottom of the lake. Therefore, we can actually calculate how much oxygen would be in the water during the spring, assuming that it's fully mixed. And now we have the total amount of oxygen that we started with, and we have our measured data, so we know the total amount of oxygen that we ended up with. The only missing piece of the puzzle is the time that has now passed from spring to summer. And luckily, we can leverage some past research on lake physical process modeling, where we can use some sort of combination of air temperature, lake surface area, and lake depth to actually estimate the day of the year where a lake would stratify. And there's a number of different, papers out here. I just showed a few that do this. So there are some options. But before we went out and kind of upscaled these methods, we wanted to test if they really worked based on our measured data. So the first thing we did was we plotted out our very high resolution every 3 days worth of data for Big Cedar Lake. So this plot here shows date of year on the bottom, our depth on the top, and instead of the squiggly lines, we now have a heat map of temperature here, where the warmer colors indicate greater water temperatures. And we can very clearly see this onset of strong summer stratification that's happening kind of mid to late May, where those surface water temperatures are really warming up and creating that disconnect between those bottom layers of water. But if we look closely, we do see, it might be hard to see on the screen here, but some periods of weak stratification occurring in the early spring, where the surface waters are just a little bit warmer than the bottom waters, but it can still create that kind of disconnect and lack of mixing between the layers. And when we use some of those past models to predict the start of stratification, the date aligns right at the end of April, and we actually see pretty good agreement between our observed data and what those models were predicting. So this was encouraging, so we decided to move forward. Okay, let's do this for the 8 lakes where we have measured data for, and let's compare our measured rates to the ones that we predict based on our single summer profile, and see how well they match up. And these are the results here. So we have on the left our 8 lakes where we have our measured dissolved oxygen depletion rates. And then on the right, we have those same dissolved oxygen depletion rates just predicted through our knowledge of the date of stratification and that relationship between temperature and dissolved oxygen solubility. And if you look at these pairwise comparisons of points, some of them are okay, some of them are pretty similar, but all in all, our predicted rates seem to be much greater than our measured rates. Greater meaning here a more negative number, so more oxygen is being depleted faster. And because of this consistent difference among all the lakes, it raised some red flags, and we really didn't think this was acceptable. There was something going on here. There's some sort of error happening. We started to kind of investigate, like what, go through the list, what could be going on here. The first thing we asked, was it stratification dates? Well, we had our Big Cedar data to kind of corroborate that, we were pretty close in terms of that estimate. Was there something weird going on with dissolved oxygen depletion where it was non-linear, maybe happening faster and slower at different points of the year? Well, we had our measured data and it showed very consistent dissolved oxygen depletion over time. So it wasn't that. So we were kind of puzzled. And that's until we decided to look at our high resolution oxygen data from Big Cedar Lake. So this is the same sort of heat map with depth, time, and instead of temperature now, we show dissolved oxygen, where the warmer colors here are lower dissolved oxygen concentrations. And right away, if you look at this figure, we see that in contrast to the fall, where the whole water column was uniform, in the spring, we have these pockets of low dissolved oxygen at the bottom of the lake, indicating that the water column is not fully mixed here. So what can happen is that heat can actually be transferred, through the ice as it's melting. We could have rapidly warming springs that are warming up those surface layers of water. And in reality, the lake might not stay uniformly mixed long enough for the oxygen that has been lost to be fully replenished and have a water column that looks like the part on the right there in the fall time. So we decided to look at this further and generate these same figures for all 8 of our lakes. The Big Cedar Lake is duplicated here. So I've separated the 8 lakes based on lake size. So these first 4 are smaller lakes. And we, pretty clearly based on these figures, we see that in May, in the spring, that there is these pockets of low dissolved oxygen at the bottom of these lakes, indicating that they were not fully mixed in the spring. If we look at our deeper water lakes, this low dissolved oxygen signal is less clear, but if you kind of zoom in and look at it a little closer, there's still less dissolved oxygen in these deep waters than we would expect based on our temperature and solubility relationships. So the reason our predicted data didn't really work out very well is because of some of these complexities in spring mixing. It turns out these lakes weren't as mixed as we thought. So we couldn't really leverage that broad scale monitoring data as we wanted to generate a larger data set of these dissolved oxygen depletion rates. What this also means is that spring mixing doesn't really follow exactly what's written in the limnology textbook. It's a little bit more complex, which further raises the question, is this insufficient spring mixing common in the Kawartha lakes? And what is driving this insufficient mixing? Is it the morphology of the lake, as we saw with our big and our small lakes? Does it have to do with weather in the springtime? Or does it have to do with the dissolved oxygen that's lost in these lakes over the winter time? So I can't really answer the first 2 questions, but I do have some data on the winter dissolved oxygen profiles. And given our Big Cedar data, where we saw that in the fall, yes, this water column was fully mixed. Our oxygen was at 100% saturation. So that's good. Given that, we can actually calculate the dissolved oxygen depletion rates over the winter, as long as we know when it was cut off from the atmosphere. For the Kawarthas in 2022, the date where the ice kind of formed, broadly was around December 27th. So based on our dissolved oxygen profiles collected in February here, we can, I calculated some of those rates, and I've plotted them next to our summer rates for comparison. So winter on the left, summer on the right. And if you ignore Raccoon and Gold Lake for now, we can see that the summer rates were all greater than our winter rates. Again, greater being negative, more dissolved oxygens being depleted. And this is as expected because the temperature in the bottom of the lakes in the summertime is warmer than in the wintertime. These warmer temperatures can promote more microbial activity and their associated dissolved oxygen depletion. As for Raccoon and Gold Lake, they are in the complete opposite direction and look really weird. I'm not really sure what's going on. I have a couple hypotheses. Raccoon Lake is very small, so it's possible that the ice formed maybe a bit earlier than the general Kawartha Lakes ice on date. In regard to Gold Lake, just due to accessibility constraints, we didn't have a snowmobile. We were walking out on the ice here to collect these samples. We actually went to a different basin to collect our winter Gold Lake profile. So there could be some spatial differences here, between basins within a lake. But these winter data are really informative because it's really the dissolved oxygen that's being lost over the winter time that needs to be replenished in the springtime in order to have that uniform, that uniform concentration of dissolved oxygen and that fully restored 100% dissolved oxygen saturation. So collecting some of this winter data can really be beneficial in the long run. So dissolved oxygen is complex, especially in the spring. This winter to spring transition here sets the fixed supply of dissolved oxygen for the summertime. So that shoulder season, so the season we're in right now, is really, really important. Some spatial variation in lake morphology seems to be linked to some of those differences in the mixing of these lakes. And it's likely that temporal variation in spring conditions and weather is probably driving some interannual variability we see in our summer dissolved oxygen concentrations. But back to our research questions, we were able to shed some light and collect some data on what are the rates of deep water dissolved oxygen depletion in the Kawarthas. But because of these complexities in the spring mixing, we weren't able to leverage the broad scale monitoring data with those singular profiles collected in August to really answer our next question and scale up our data set as to what are the factors that drive differences in dissolved oxygen depletion among lakes. And we weren't able to answer the question of have these rates changed over time. So like I said, these vertical profiles have been common in limnology for a while. Might be kind of hard to see on this map, but this is a map from 1969. And in the top left corner, there's actually a temperature and oxygen profile data for Gold Lake. So the hope was that we could extract some of this past data to look at how things have changed, but it doesn't seem to be the case. So in order to answer these two questions, we really need more targeted monitoring. But we were able to provide some insight on our next question, which was what are the ecological consequences of dissolved oxygen depletion? And one of the things we could shed some light on is habitat availability. So the Canadian Water Quality Guidelines for the Protection of Aquatic Life state that the lowest acceptable dissolved oxygen concentration for freshwaters is 5.5 milligrams of oxygen per liter. And based on our broad scale monitoring data of those profiles collected in August during the summer, we can actually pair that with our geographic data on lake morphology to calculate the proportion of a lake's volume that falls below this guideline value. And we did that for about 35 of our Kawartha Highlands lakes here, and the results are shown kind of summarized on this box plot. And if we look at this box plot, we see that there's a lot of lakes that are on that zero line. So this is a good thing. The whole water column is well oxygenated. There's no loss of habitat due to oxygen depletion. But if we start to look up towards the top of our graph here, we do see that some of these lakes have greater than 50% of their lake volume falling below this guideline value and would be unsuitable for freshwater life. If we kind of look at everything overall, altogether, on average, approximately 50% of the Kawartha lakes that we sampled have greater than 20% of its lake volume that would be unsuitable for aquatic life. So I really didn't know what these values were, but I thought that was pretty high. So oxygen is needed, for fish to breathe. We know that, and we know that the loss of these habitats can affect fish and other organisms. We wanted to take that one step further in our Stony Lake project in partnership with Graham Raby's lab to look at how water quality and low dissolved oxygen can actually affect the movement and behavior of fish through fish tracking and acoustic underwater telemetry. This work is very much in progress, but we can see this animation of this map here from some of our preliminary work last summer, where we can actually map out dissolved oxygen and other variables in 3D across the entirety of the lake to identify different spatial locations where we might, see like low dissolved oxygen, like in Clear Lake there, and maybe there's some avoidance of those areas due to fish. So we can begin to explore those relationships in a little more detail. The other consequence of dissolved oxygen depletion that we have been investigating is related to biogeochemistry. I've put Kate's photo on the slide here. She's a PhD student at Trent. Some of you might know her through some of the lake sampling work that she does. But she's investigating how the low dissolved oxygen at the bottom of lakes can actually result in internal phosphorus release from lake sediments increase nutrient availability in the water column and stimulate phytoplankton growth. She's finishing up her PhD this summer. So maybe at one of the next meetings, you can recruit her to share her findings. And finally, the last question that we wanted to shed some light on is dissolved oxygen depletion a concern regarding the sustainability of the Kawartha lakes and all the things that we think are important about our lakes? And the answer I have here is probably, but we are data deficient. Moving forward, there's a need for an increased understanding of dissolved oxygen depletion to help safeguard these lakes, especially the ones where we have these high dissolved oxygen habitats. And like Bob said at the beginning of our meeting here, we really need to collect these data to establish baselines in order to make future comparisons. And ultimately, without collecting this data, we have no idea about the changing status of these ecosystems. So the good news is this picture was from, I think, 2 days ago. We've been out on the lakes already collecting some spring dissolved oxygen profiles. Since we couldn't leverage our routine monitoring data, we're going to put out a greater effort to hit more lakes and calculate these dissolved oxygen depletion rates on a larger scale. So stay tuned for that. But I do think there is a genuine cause for concern regarding dissolved oxygen depletion, in particular in relation to climate change. So with climate change, we're expected to see increasing air temperatures. Those increasing air temperatures will increase surface water temperatures, and those increases in surface water temperatures will increase the duration of stratification for lakes. So the period of time where those bottom waters are cut off from the atmosphere. So this graph here is just I'm sure you've seen some of these before, but a climate change forecasting graph where we have year on the x-axis, and then we have stratification duration on the y-axis for a number of different climate change scenarios. And we can see these sharp increases especially in the more scary climate change scenarios where this stratification duration can almost double. And if that duration increases, just by the nature of those bottom waters being cut off from the atmosphere for longer, more oxygen will be depleted and be cause for concern. The other thing that's a cause for concern regarding climate change is due to changes in lake ice phenology. There's some evidence from some Wisconsin lakes that earlier ice off can result in warmer water temperatures at the bottom of lakes. With the warmer water temperatures, that water can hold less dissolved oxygen initially. Those warmer temperatures also promote microbial activity and associated dissolved oxygen depletion. And if we just focus on the trout lake data on the bottom of this figure, we can see that in early ice off years in red, that the water temperatures at the bottom of Trout Lake here were a couple degrees warmer than in late ice off years in blue. And as a result, the oxygen in that lake went below 40% dissolved oxygen saturation much earlier in the year during those early ice off years than in the late ice off years. And to bring it back to this graph, if these physical changes in lakes, with warming water temperatures and stratification and changes in lake ice phenology, we may actually see this deep water temperature trend move into the positives. And just like the surface waters, if we see the temperatures of these deep waters increasing, they can hold less oxygen. So we'd expect to see our oxygen concentration trend to further increase, resulting in more cause for concern and making situations worse. But I'd like to bring it back to this map here and remind you that the Kawartha Lakes are these really high importance ecosystems and generally good at ecological standing, but they are highly threatened. So effective ecological monitoring is fundamentally important here. without, especially without the data that the Kawartha Lake Stewards and all these partnerships have been collecting, we'd have no idea about the changing status of these ecosystems. So, you know, I'm always happy when I come to these meetings because it's you know, all the work you're doing is super great. So I just like to applaud you for that. And thank you for giving me the opportunity to share some of my research today. And if you have any questions, I'll be happy to take them, or you can take a picture of my email and I can get back to you.
Thank you so much, Nolan. We're running quite short of time. So what I'd suggest, we're not going anywhere. We're just running a little short of time on our spring meeting. So what I would suggest, if you want, Nolan's going to hang around. You can chat with him. And if you have any questions, don't forget, I think Jeff's still here and his boat is still out there. And all of your board, all of us who work for you, work with you, we're sticking around. So have another cup of coffee, have another Timbit. I can't go home with any of those Timbits or I'll eat them. And thank you so much for being here. Thank you to Nolan for that great talk. Always love hearing about, I mean, the folks at Trent, Paul and others, and Maggie and others, they not only can buy neat stuff like that profiler, but they get resources like this guy, you know, training his great research eye on questions as we heard about today. So thank you very much for that, Nolan. And again, thanks to you folks for coming, and we'll see you at the AGM or we'll see you at the lake. Cheers.